All right, welcome back to our discussion on meiosis. Now, as we're going to talk about meiosis, we're going to skip the sources of information here for a little bit. We have to talk about life cycles of organisms and kind of where meiosis fits into the life cycle of an organism. You know that mitosis plays a big role even from when you're a zygote. You're going to go through those cell divisions simply to be able to grow and create identical copies of cells. So anytime we have growth, that's going to be mitosis. But meiosis is going to be very significant, as you can tell here in this slide, for reproduction and allowing a species to continue, especially sexually reproducing organisms. So if we looked at a diagram of a human life cycle, we know that from zygote, we're going to have the process of mitosis because that's going to be causing growth. However, when we want to continue the species, the male and female of the species need to create the sex cells, the gametes. That's where meiosis comes in, because meiosis needs to cut the chromosome number in half so that when a sperm meets an egg, we get back to that proper diploid number of chromosomes. Remember that we're going to call 2n. And so meiosis is a process of cutting that chromosome number in half to form what we call haploid cells. So the sex cells, the gametes, the egg, the sperm in humans are only going to have half the number of chromosomes, which would be 23. All right, so guys, here's a slide that's kind of talking about some of these terms. The process of meiosis is to create specialized cells, the gametes, sperm and egg. They're typical diploid cells. The full number of chromosomes are what we call the somatic cells, the typical cells of the body. All the cells except for the gametes, the eggs and sperm. And there we see that 2n representation where n is the haploid number. So eggs and sperm are going to be haploid. They have only 1/2 of the full complement of chromosomes. Right? And of course, meiosis creates variety. Different is good. Remember that mitosis is creating identical cells, no variation. But we want variation with sexually reproducing organisms. And so that's meiosis is going to create variety. So sexually reproduction gives us that variety we're looking for because we're combining two sets of genetic information. All right, so two processes occur in meiosis that ensure variety and diversity that don't occur in mitosis, and we're going to be looking at this in some detail. The first event is crossing over, and the second event is random assortment. Not going to say too much about that now until we actually get into the phases or stages of meiosis. This is an overview, guys, and of meiosis. You'll notice it has the same kind of phases as mitosis does. There's just more of them. So remember, we are going to start with an interphase. So that's true for meiosis as well as mitosis. And everything we say about interphase for mitosis holds for meiosis. We're going to have some growth. We're going to have some doubling of the genetic information, right? And then we move into the exact same phases, PMAT, remember, prophase, metaphase, anaphase, and telophase, but we're actually do it twice in meiosis. So we say that we have a meiosis 1, so PMAT is going to have a 1 after it, and then we have meiosis 2, and we're going to have the Roman numeral 2 for the names of those phases, prophase 2, metaphase 2, etc. So let's take a look at meiosis 1. Meiosis 1, guys, is the actual division phase. It's actually where we're going to cut the chromosome number in half. You're going to see a lot of similar terminology. So remember, in meiosis 1, we're going to start with prophase 1. And a lot of the similar things happen as in mitosis. We're going to see a disappearance of the nuclear membrane. We're going to see a condensing of the chromatin to form individual chromosomes. We're going to see those centrioles again, and the centrioles are going to go to the poles. They're going to create their spindle fibers. But now here's what's different, and we're going to see individual slides on this in a moment. The chromosomes, of which humans would have 46, they're going to, they're actually 23 pairs, and they're going to come together as what we call the homologous pairs. Now, why do we have pairs? Because we have one chromosome from mom, and we have one chromosome from dad, right? And so they're similar in size, shape, and gene arrangement, and in prophase 1, they're going to find their pair. They're going to find their match and make a pair. We call that homologous pair. And they actually even intertwine, and then they're going to do this thing called crossing over, which is actually going to have some DNA segments are going to trade places with the inside sister chromatids. We often call this a tetrad because technically we have 4 sisters pretty close together there, right? Okay. And then we're going to move into metaphase 1, homologous pairs. Now, notice, guys, this is different than mitosis. The pairs align on the equatorial plate, not as a single not in single file like they do in mitosis. The homologous pairs line up, and then the pairs are going to be separated in anaphase, and then we are going to go through a telophase 1 where we're going to create 2 separate cells. Notice we were going to have half the number of chromosomes. So we are going to look at it in a little more detail coming up. So in meiosis 2, if we continue, remember we created those 2 cells, now with just 2 chromosomes each. They're going to go through meiosis 2, which looks pretty much exactly like mitosis. There's really very little difference here, guys, because we end up with 2 cells with 2 chromosomes, just like this if this was considered to be the parent. But remember that we've done this twice, and we actually started with 4 chromosomes. So at the end of meiosis 2, we have 4 cells with half the number of chromosomes in each nucleus, which is 2. And notice the variety. Neither of these cells are genetically identical to any other. So we've created that variety. So again, if we go through meiosis 1 a little more slowly, we've said meiosis 1 is the reduction division where we actually cut the chromosome number in half. We start with a diploid cell, but in meiosis 1, that's when we cut the chromosome number in half. We simply maintain that chromosome number in meiosis 2. But notice we have 4 cells when we're done, not 2 like in mitosis. So in prophase 1, we have a lot of those events that we already talked about. Nuclear membrane dissolves, the centrioles go to the poles, we get those spindle fibers, they attach to the centromeres of the chromosomes, which we can now see. Remember we said that the chromosomes are going to come together to form pairs. By the way, the term for that is synapsis, to get close together. There's one that's from mom, maternal, one that's from dad, that's paternal. But one homologous pair is equal to 2 chromosomes. So remember we have 4 sister chromatids, and we can call that a tetrad. So again, we can see it visually here. Remember that there's in interphase we double each chromosome. So there is one homolog, and there's the other one, and they're going to come together. Synapse. And then cross over. So here's the visual of the crossing over. Whoops. All right. So, and guys, crossing over is a random event. It doesn't always happen. It doesn't have to happen. And if it's going to happen, it doesn't have to happen in the same places. But it is going to happen where the sister chromatids, the inside sisters, sort of intertwine between the mother and father chromosomes. Okay? There are definitely different chromatids, 4 different chromatids when crossing over is done, right? 4. This one is different from this one, which is different from this one, which is different than this one. All right? So now the sisters are no longer identical at all, like mitosis. So the true result of crossing over, guys, is variety in every gamete. All right? Every gamete will be different because of crossing over and random assortment, which is coming up here in a second. Now in metaphase 1, remember that the chromosomes move to the equatorial plate by those centrioles. Okay. In meiosis, spindle fibers from one pole attach to a member of the homologous pair, while spindles from the other pole attaches to the other member of the pair, right? So instead of there just being one centromere, in mitosis, we actually have 2 centromeres because we have those 2 homologous pairs. Now they're not in single file. Remember we said that they're actually lining up on the equatorial plate as pairs, not 46 in a line. So here we are right here, homologous pairs lined up on the equatorial plate. And in anaphase, they're going to be separated. But before they're separated, we need to talk about random assortment. The way the pairs line up on the equatorial plate is random. So in other words, remember we have a mother and father homologs together. All the maternal homologs don't line up on one side, like in the gym. If we said all the boys line up on one side of the gym and all the girls line up on the other side, it doesn't work that way. It's as if you had a boy-girl pair, but you said, okay, line up in the center of the gym, but randomly. You get to decide whether the boy's on one side or the girl's on one side. So that's what happens in metaphase 1 along the equatorial plate. And that, since that's a completely random event, that's another way that we get a lot of variety in the final gametes or sex cells. Okay, it's completely random the way they pair up. Okay, so down here we could have had this red chromosome, the blues on this side. Remember, all the sisters are different. And then over here we have the blue on this side, on top, and the red on the bottom. But we could have just as easily had the red on the bottom over here and the blue on the top. So that's called random assortment. And so, yes, just imagine the possibilities. If you did the mathematics, it results into 2n, where n is the number of haploid chromosomes. So since we have 23 is our haploid number, there's 8 million, approximately 400,000 different possible outcomes of how the cells could line up on the equatorial plate. And then remember, there's crossing over that makes all those sisters very different from one another. So that's why we don't really have 2 people that are, except for identical twins, being genetically similar or identical. The odds are so stacked against it. Now, in anaphase 1, quite quickly, we get the separation of the homologous pairs, right? And we call that segregation, separation. The sister chromatids are going to remain intact for each of the homologous chromosomes. So no centromeres actually split this time, because remember that the spindle fibers, one grabs one of the male centromere, and the other spindle fiber on the other side grabs the female centromere. So we actually just pull the homologous chromosomes apart. And in telophase 1, pretty much everything that happens, except we don't have chromosomes going back into chromatin. They stay as thickened chromosomes. But we don't get our nuclear membrane back, I don't believe. Oh, actually, I think we do. So I'll change. Nuclear membrane does reappear, but we don't go through a separate interphase, guys. No interphase. Okay? And no, those 2 cells that we've created at this point at telophase 1 are not genetically identical. Now we have no more crossing over or anything happening like that. Okay, just going to go through I just want to get to meiosis 2 quickly here. You can have a quick look if you go through the slideshow slowly if you want to read those questions and answers. So now we're going to pick up from here, guys, because we've got our 2 cells. So now we're going to move through meiosis 2. All right, this is the overview of meiosis 2. We've seen, I think we've seen this already. We're picking up right here with prophase 2. We're going to get our centrioles back. We're going to go to the poles. We're going to have our spindle fibers. And it's similar to mitosis. The chromosomes are there. They're going to line up in single file this time. In metaphase 2, the centromeres are going to split, just like in anaphase of mitosis, but this is anaphase 2. The sister chromatids get pulled to the poles. They're single-stranded at this time. And then, of course, we're going to get our nucleus forming in telophase 2, and we're going to get another cytokinesis to separate those 2 cells into 4. All right? So again, this is just review of things we've talked about before in terms of haploid. Okay. Versus mitosis, remember, 4 haploid cells for meiosis, 2 diploid identical cells if it's mitosis. Just a few more. This is just another artist's rendering, guys. Remember, we go through interphase, doubling the genetic information, formation in prophase of the homologous chromosomes, line up on them. Now, and we've had crossing over. Crossing over actually happens more in prophase and metaphase, not in anaphase. All right. So that's it. And now the next time I talk to you guys, we'll get to specific gametogenesis, formation of the gametes, which of course is what meiosis does. But we're going to look at it specifically for humans in the formation of sperm and in the formation of an ova or ovum.